I am here to talk about a 1.5-year statewide survey that we've been working on with our NDSU collaborators. So where are we going? Show of hands in the room, who has not heard of NAGC? We're that popular? I only see like 2 or 3, have not. Okay, well, you can ignore the first like 5 slides here because I'm going to introduce ourselves, who we are, and where we fit in the wild world of weeds. Then I'm going to talk about the statewide survey, the first year of results, briefly touch on the upcoming projects that we have going on, and then some begging at you because we want to grow this statewide survey. All right, so the place, I'm going to just kind of skim through this. We are on campus. We kind of have this Russian nesting doll situation where we're on campus, we're in a USDA building, but we are our own facility. We are a private, not-for-profit organization. Here are the people. This is us trying to fit in as growers. So this is our best flannel that we have as lab personnel. We are an ISO 17025 accredited laboratory. There's 7 of us full-time that work at the lab. We have a lot of things. Our things are a piece of equipment, genetic analysis work. We kind of get bored in the lab, so we start naming things. So pick your favorite 90s or 2000 sitcom or movie. We probably have a machine named after one of those. And so again, we're a high throughput testing facility. And so we have the ability to test upwards to thousands of samples in a given week. And we play in a space that looks something like this. We play with test tubes, essentially. So each one of those wells can be a single test, which could be looking at a single genotype for a particular weed species. It could be looking at 3 different targets if we're talking about pathogens, which is not why we're here today. But we're thinking in this laboratory space. And so our general approach as a laboratory is to collaborate. We want to complement and not compete. So if we know of another diagnostic lab that's providing a service and they're doing a great job, we move on and find something else to do with our time. And we also want to provide this private confidential reporting to clients as well. And so that's the growers, that's agronomists, whoever uses our diagnostic support. When it comes to our weed-focused approach, I like bothering all the weed scientists at NDSU. I go up to Joe and say, hey, what do you got growing in that greenhouse? Is there any leaf samples that need genotyping? Hey, Brian, over in Minot, do you have anything growing in your greenhouse? Because again, we're just a lab. We don't have our own field. We don't have our own greenhouse space. And so we really rely on this collaborative environment. We want to increase the access to DNA technology to everyone. It's expensive and it's very technical. And so we want to be able to provide that to growers as well as agronomists. And then also provide fast turnaround time. So if there's particular tests that have the ability to help with management, we want to be able to provide that in a timely manner. All right, so what do we test for? In weeds, we test for target site mutations or the variants that leads to herbicide resistance. And speaking for myself as a geneticist, maybe Dr. Christophers feels the same way, but we view the world a little bit differently. Here you might see soybeans with water hemp. I see a bunch of genomes out there in that field. So I see genetic variation. I see vehicles of genes and genomics. Same within the greenhouse. So you're looking at greenhouse plants, cute little weeds there. I'm seeing double helices. I'm seeing DNA. I'm seeing the ability for us to answer some questions based off of the genotypes and the genetics of these plants. And so I wanted to kind of briefly do an overview of the mechanism that we focus on as a lab. So take this plant. Like I said, geneticists are weird. We see a plant. We also see DNA. And so within this genome, there's a gene that codes for a protein. This protein is going to be an enzyme. Enzymes help speed the reaction of some sort of biochemical process in the plant. And so essentially what the enzyme's doing is it's speeding up this process that takes a substrate and breaks them down into products. These products could be essential amino acids necessary for plant life. And so this whole process makes a happy weed. However, if you include a herbicide as a competitor, what it does is it locks down that enzyme, and then you get a buildup of substrate. This buildup of substrate tends to be toxic to the plant, and you get a sad plant. They can stress out and it can die, essentially. So what we're noticing then is that you have a wild type, or what you could probably consider a normal enzyme, that has changed. It's changed in its shape. But say in these weeds that I'm showing here, that in the gray, this enzyme is still functional. It's got a mutation potentially, but it's still functional. That substrate, even in the presence of the herbicides, can still make those products and still be an okay or a happy weed. And so we know that these enzymes change shape due to a mutation. So we can look in the genome and find that one site. For example, this G turns into a C here. That's all it takes for a target site mutation to lead to herbicide resistance. And so as a diagnostic lab, what we can do is develop a test with kind of two colored probes. One will tell us if it's a susceptible plant. The second one will tell us if it's a resistant plant or a plant that has that mutation. So that's where we come in with the goals of the statewide survey. We want to seek these freaks. We want to seek these weeds that have that mutation or that variant that leads to herbicide resistance. And we can do that by that genotyping test. We wanted to make sure we could make this survey easy for those that want to collect and submit to us. We also want to have fast reporting for early detection. I think one thing that we learned throughout today is that when you're spraying these weeds, they need to be small, right? They have to be about 1 inch to 2 inches. We can do the test on a 1 to 2 inch plant. And we wanted to develop county level maps for regional trends. The whole point of doing a survey is to find out, okay, where are these genetic variants and how we can use it to explain some of the patterns that growers are seeing with herbicide resistance. All right, so with any study, there's limitations. The first limitation is that these tests are very specific. They only target known genetic markers linked or associated with resistance. What that means is that the test itself, the ones that we use, do not detect new mutations. We have to develop another test for those. And secondly, it cannot detect non-target site mechanisms. And I think Dr. Lancaster went over that during her keynote. These non-target site mechanisms, as a geneticist, we don't worry about those when we're dealing with this test. They are obviously important. And so we've built this partnership with the Weed Diagnostic Program already going on at NDSU. So they've been growing up all these plants in the greenhouse and doing the herbicide-resistant trials. And so we come in and do the genetic testing for these target site mechanisms. The great thing about the genetic testing is that we can test a lot of samples quickly. They can be single seed sources. They can be leaf samples from actively growing plants. And so we're able to do a lot more testing, at least survey-wise, than you can in the greenhouse. Because the greenhouse, typically those trials are going on the off-season only. It takes several months to grow up the seed, to do all the spraying. Obviously very important work, but the genetic work helps supplement what they're doing in the greenhouse. And so we have support through North Dakota Soybean Council for this survey. It kicked off in August 2023. So Joe and I went out to a field to make these videos on how to collect for the survey. And you know, when you're in a video, you know, sometimes it's good to kind of see kind of your mannerisms and see what you look like when you're out there doing this video. And so if you zoom in on my face, I have two theories for this face. I might be a little pissed off, maybe a little confused. I have two theories why I look like this. One, this is the face a lab geek has when he goes out to the field and feels out of place. 2, I might be disgusted because I think this is Joe's soybean field. I was disgusted with how many weeds were out there. He is the worst soybean farmer I've ever seen. So that might be that phase 2. We're not sure. Stay tuned. So the survey focused on 2 weeds, pigweeds, kind of that family, really focusing on water hemp, and then kochia. Current tests now detect only certain DNA markers associated with resistance. So group 2, 9, and 14. I think of them in terms of the groups or the site of actions. I was told I should probably put some brand names up as well. So there they are for you to kind of gauge where we're at with this testing. All right, so the collection is easy. You've seen this a couple times already through various talks, but all we need is a couple leaf samples from a single individual. The test itself, we're only doing a paper hole size punch leaf to test all 3 of those markers on these plants. Easy submission, so once you have collected 4 samples from a field or from multiple fields, you shove them in a prepaid envelope and a submission form, and they arrive to our lab in about 2 to 3 days. And then the submitter themselves get their own confidential reports. And so we don't share this information specifically with anyone besides the submitter. This right here, you can see is Doogie Howser's report. There's a couple laughs out there. Some people know who Doogie Howser is. Oh, I'm aging myself. Moving on. So the testing is free year-round. Obviously, you're not going to be testing right now because there's snow on the ground. But the turnaround is 7 days or fewer or less. And essentially, it takes a couple days for us to get the envelope. We provide the testing, and then we send out that report. I think most of the time during the busy season, so in the early spring, we're usually reporting out results within 5 business days. All right, so we're here for the results over the last year and a half. Note that this is the genetic results only. I know there's a lot of greenhouse work going on, so I might be missing counties based off of the greenhouse results. I think all of us great minds need to get together and probably combine all the greenhouse and genetic data into a county level map. So that will probably be coming shortly. But this is all only just the genetic results. All right, so we received 520 samples through the statewide survey. That's 80% of the counties here. So kochia, we received them in counties only in blue. Pigweeds in counties only yellow. And if we received both of those groups or species, those are the green counties. There was a couple ways to show these data. I know you've been hit with a lot of information, a lot of numbers and figures today. I'm going to pile on that a little bit here. But what I did is I split it into the 2 groups, so the pigweed group and kochia. Pigweeds, again, are mostly water hemp samples, but there is redroot and Powell amaranth in this group too. But we received pigweeds from 21 counties, and it equates to 79 plants. And so here I'm showing you in the color the percentage of plants, the 79 individual pigweeds that we received that had the resistant marker. So for example, in group 2, the ALS inhibitors, 33% of the pigweeds we received had that marker. The group 9 and group 14, those percentages are based only on the water hemp samples. Those markers really only work on water hemp and Palmer amaranth. So these data are a little skewed. Group 2 is including all of the pigweeds. Group 9 and 14 are only the water hemp samples that we received. All right, the next few slides are heat maps. So essentially what I'm going to do is run through all the 3 groups that we test for, their resistant markers. And if we received a sample from that particular county in the state, you'll see a number. If it's 0, the county is going to remain white. If it's one of the higher numbers where we saw the resistant marker, it's going to be the darker color. So in this heat map here, the most samples that we received from a particular county was 6 weeds. And so this county way up here, 6 out of 6, all of the pigweeds that we received from that county had the resistant marker. So I'm just going to kind of go through the next one. So this is group 2 marker. This is the glyphosate-resistant marker, so group 9. And then group 14. So if you want to see that all again, pick your favorite county, maybe the county you live in. If you live in a county that doesn't have a number, you should feel bad that you didn't submit samples to me. But you can see most of the samples came from the eastern side of the state. And so if we kind of combine all that data and say, okay, within a county, what counties did we see all 3 resistant markers in? That's those in pink here. So it's 29% of the counties that we actually received pigweed samples from. So the ones that we received pigweed samples from, but we didn't find all 3 resistant markers in, are gray on this map. So again, we didn't really have a lot of pigweeds to say a lot about the county level stuff. Now, that was the population-wide or county level. What we can do with genetic testing, though, is see how many had the stacked resistance. All we needed was that one leaf sample, and we can test for all 3 markers in that same plant. And so that's what this stacked bar graph is showing you here. So for pigweeds, 48% of them did not have a single resistant marker in them. 37% of pigweeds that we tested had one way, most likely the ALS marker. There was 11% of the pigweeds had two-way, and then there was 3 individual plants that had markers linked to resistance to all 3 of those herbicides. And so those 3 plants were found in these 2 counties right here. The numbers illustrating the number of plants that we found. So again, we want to kind of build up more of the pigweed testing. So if you have large problems in your county with pigweeds, please participate in the survey this next year. All right, moving on to kochia. So kochia, we have more counties and way more plants. There seemed to be way more interest in that for the statewide survey. I'm showing you the data the exact same way. So again, these are the resistant plants by herbicide group. So we had over half of the kochia samples we received have that group 9 resistant marker. Group 14, again, that's the new one that you've heard of, the new issue that we're seeing in the state. We do have a genetic marker linked to that resistance, and so we had 40% of the kochia that we tested had that resistant marker. Here again are the heat maps for the state. Again, pick your favorite county as I kind of go through these. You can see the southwestern side of the state is winning. I have to make a trophy or something, I don't know. Try to battle out all these different regions in North Dakota so we can get more samples. But there was a lot of testing down in the southwestern side of the state. And so that is also leading to kind of more results there. It's not necessarily there's more of an issue on the western side of the state. It's that we got more samples and we can say more about that side of the state. And then this is group 14, so this is that new marker. And so I think what was concerning is that we were not only seeing it on the west side of the state, but we were seeing it also on the eastern side of the state. So just to kind of, So just to kind of show them again for those of you that want to see your favorite county. And so if we, again, combine all that together at the county level, 59% of the counties that we tested had kochia with all 3 resistant markers. Did it also within an individual plant. And so again, we're just seeing more within individual stacked resistance. So 7% had all 3 resistant markers in it. That equated to about 33 of the kochia samples that we received last year. And so this is their distribution. So these are the ones where we found 3-way resistant markers in those kochia. So again, some of that has to do with how the samples were collected and where they were from. We got a lot of samples from the western side of the state, so we're seeing a lot of that 3-way resistance in this survey. So some takeaways: widespread distribution of HR genotypes. That's probably not that surprising to most of you that deal with herbicide-resistant weeds on a daily basis. We found that group 14 resistant marker in 24 counties that we tested. We're seeing these stacked herbicide-resistant genotypes, mostly in kochia. And again, hopefully you understand that the survey provides an early detection system. we're able to test those plants when they're really tiny and you're first seeing them out in the field. We want to expand this, and so we have future tests in 2 weedy grasses. So we're looking at wild oat and green foxtail. We're looking at group 1 and group 2 markers for a couple reasons. The first most is that in the literature, there's a close linkage between mutations that have already been found that we can develop tests for. We've also surveyed a few green foxtail and wild oat from North Dakota, and we're finding those target site variants in the state. So we know it's going to be a useful test in the future. But we still need your help. This is where the begging comes in. So really the survey success depends on volunteers like you out in the audience. And so these kits are available at North Dakota County Extension offices throughout the state. I'm going to send out more probably in March. I didn't bring any today because I didn't want you to lose them. I want you to get them and use them as soon as possible. So I'm going to send those out in early March to all the extension offices. But you can request your own. If you want a couple of collection envelopes yourself, I am happy to mail them out to you as well. And some of you might not be from North Dakota, and so this testing is available to everyone, but there's fees associated if you're not submitting in North Dakota. And that's because we have some great supporters and funders. All of this program is funded through the North Dakota Soybean Council and North Dakota Corn Utilization Council, and we're getting money also through the Department of Agriculture as well. So what I'm going to have is I'm going to have a little collection bin, and if you have your business card and you want some collection kits, you can pop your business card in there or a slip of paper with your email and name, and I will reach out to you when we're ready to send out those kits. So with that, I'm happy to take any of your questions. Thank you. Yeah, so the question is that when we're doing this testing within pigweeds, that's a family, and there's, red root, there's polyamaranth, there's water hemp, and where's that distinction? So when it comes to the genetic testing, we rely on what's out there in the literature, and there really hasn't been a lot of target site mechanisms identified in those monoecious pigweeds for glyphosate resistance and PPO inhibitor resistance. For group 2, it can be the same mutation across all those individual species. And so we can use the same test, the same genetic test, regardless of what species it is, because it's a conserved area and the mutation is the same across the species. The question was, what about Canadian thistle and other types of potential noxious weed species? Yeah, there has been I don't know about that particular species, but across species, if you go to, weedscience.org, the large one, they have a list of all the mutations, and these sites are pretty much the ones that you see across these species. So test development is possible for sure, and it just comes down to funding and resources and all that. Yeah, so the question is, how reliable is just going based off of the genotyping data when we know that phenotypically we see a lot more of that out there? That is why we have this perfect little footer in our testing report that says, look, we're looking at a very small window of what leads to resistance when it comes to group 2, and that there are other mechanisms out there. And then we highly suggest that if you get what we call a wild type or susceptible report, that you don't take that at face value, that it's probably best to do more greenhouse results or what you're seeing out in the field. Sure. So the question is, how should we be sampling fields? Should we do really intense population or patches of them? Does that help us with this information, or should we do a wider scope? For us, in terms of just getting a baseline, because that's what this first year is getting as many samples from across the state as possible, but recognizing it's a moving target, because you can introduce new genotypes into a field just through new crop seed. So it's job security when it comes to this testing, because it's going to be a moving target constantly. But we want to just get more people knowing about this survey and contributing to it so we can start seeing these regional patterns unfold.
Question online, do you want to read this one out loud?
Question online says, when will the group 1 and group 2 testing be available? I'm guessing that's for wild oat and green foxtail. Ah, man, that's a tough question to answer. We just started getting funding. It's probably not going to be until next, so it would be probably starting in 2026. But I'm doing sequencing now, so I'm getting a lot of greenhouse seed and leaf material from Brian Jenks. I think Kirk Howitt is going to give us some too, or else I'm going to ask him for some. But we're gaining information so we can develop that test. So it might be not until 2026.